Assalamualaikum and hi. So today I'm going to present to you the definition of euploidy. Euploidy is the change in the whole set of chromosomes. It occurs when a set of chromosomes did not separate during gametogenesis. It's common in plant than in animal. Gamete fusion will produce a cell or organism which have more than two sets of chromosomes. These are called as polyploid. So that's all from me.
Two types of polyploidy, which is autopolyploidy and allopolyploidy. Autopolyploidy is a condition to produce autopolyploids with more than two sets of chromosomes derived from a single species.
The second one is allopolyploidy. Allopolyploidy is a condition to produce allopolyploids with more than two sets of chromosomes from two or more species.
Let's take a look about allopolyploidy. Allopolyploidy is a condition that produces alloploid with multiple sets of chromosomes from more than one species. F1 hybrid produced from different species are sterile. We call the hybrid because the occurrence of genetic mixing. Sterile hybrid can become fertile by two methods. The first one is doubling its chromosome number and self-fertilize, and the second one is fertilizing with a normal gamete.
So the example of chromosome number and self-fertilize and also a normal gamete will be discussed next.
Okay, now let's look at first example of hybrid doubles its chromosome number to become fertile and self-fertilize. Let's say if we have species A to n equal to 6 while species B to n equal to 4. When meiosis occurs to produce normal gametes, species A will produce n equal to 3 while B produce n equal to 2. When fertilization is occur between gametes, it will produce sterile hybrid n plus n equal to 5. It is called sterile hybrid because two sets of chromosomes cannot sequence normally during meiosis. Then, sterile hybrid will undergo doubling chromosome and produce new species allopolyploidy. Finally, homologous chromosome can pair. Next, we will look how the hybrid forms an aridous gamete and fertilized with a normal gamete. The first was a hybridization between incomplete AA and one with BB producing a sterile hybrid AB. The sterile hybrid AB somehow undergo chromosome doubling. This came about by non-disjunction of all the homologous chromosomes. A new species emerged with AABB is formed. This new species is fertile because each chromosome can now pair out with its homologous partner during meiosis. A second hybridization between emerald AABB and wild-grace T-Tauchi-DD gave rise to a sterile hybrid ABD, which also undergo chromosome doubling, producing the hexaploid with T-Istivum AABB-DD. Thus, the bred wheat has chromosome derived from three ancestral species.
So next is autopolyploidy. What is autopolyploidy? Autopolyploidy is polyploids that have multiples of the haploid number of chromosome, for example, 3n, 4n, and 5n. Many plants important for agriculture are the products of polyploidy. For example, oats, cotton, potatoes, tobacco, and wheat are polyploid. So, the genome is 3n. And the autopolyploid for 3n is triploid, for 4n is tetraploid, 5n is pentaploid, 6n is hexaploid, 8n is octaploid, and for 10n is decaploid. What is examples of polyploid crops? Example of polyploid crops are for triploid crops, banana, apple, ginger, watermelon, and citrus. For tetraploid crops is potato, durum, wheat, maize, cotton, and tobacco. For hexaploid crops is chrysanthemum, bread, wheat, and oats, while for octaploid crops are strawberry, dahlia, and sugarcane. Next is formation of autopolyploidy. There is three types of formation of autopolyploidy. The first one is An unreduced gamete fertilized with another unreduced gamete from diploid cells, which is an diploid cell will fertilize with an diploid cell, or we call it as self-fertilization between two unreduced gametes from a diploid cell. We can see how it works from this diagram. And it will produce fertile tetraploid offspring genotype. Next is an unreduced gamete fertilized with a normal gamete from diploid cells, which is chromosome doubling is induced experimentally on zygotes by use of chemicals, which is diploid cells will fertilize with haploid cell. We can look from this diagram how it works, which is fertilization between an unreduced gamete with a normal gamete from two diploid cells. Lastly, the formation of autopolyploidy is by spontaneous chromosome doubling due to spindle failure during meiosis, which is giving unreduced gametes. Zygotes becomes a tetraploid having more than two chromosome sets. The karyotype of parent species will have diploid cell, which is 6 chromosomes. Unreduced gamete with 6 chromosomes will have self-fertilization and will occur zygotes, which is autopolyploid. Offspring with polyploid karyotypes may be viable and self-fertile. This is the diagram for normal diploid cells that undergo induced mutations. So lastly, a mutant. A mutant tetraploid cannot interbreed with diploid plants of the original population because it will produce a sterile triploid. When mutant fertilized with wild types, it will produce sterile. The chromosome is not paired, meiosis cannot occur. Fertile hybrids are viable and can self-fertilize but are reproductively isolated from its original population, which is cannot interbreed with its parent population. When mutant fertilized with wild type, it will produce 4N, which is the chromosomes are in pairs. Meiosis can be carried out, which is synapses can occur to form gametes. So that's all from us. Thank you for watching.